In this lecture, we're going to take a look at some of the mechanisms and what's going on when DNA is being used within cells. And if we take a look at this, we can see that your DNA has to get used between generations of cells, and that's where we look at the mechanism of DNA replication. We can also look at the DNA and what it's actively being used in within those cells here to code for certain molecules that are going to be used in protein synthesis. And then the last way we can see is DNA can actually be shuttled between cells of the same generation. But if you go back and take a look at Watson and Crick, they discovered the structure of DNA, but they actually had some problems figuring out the mechanism for how DNA replication worked. And a few people came up with some models on how it could work. They're known as conservative, semi-conservative, and dispersive DNA replication. Conservative DNA replication, you basically are going to end up where the strands in the DNA are conserved in one set of DNA molecule that is produced, and the other set of DNA is completely new. And so if you look at this, you're always going to have your old original parent DNA coming back together with the two strands, and the new DNA molecules are always going to be synthesized with completely new pieces there. Semi-conservative is a little bit different. Here you save half of the old DNA and then you make a new DNA. So you end up with half and half DNA, half old and half new. The dispersive model one is basically sort of the anarchy model of DNA replication, where the DNA can be copied wherever it wants to. And here you can see they're doing both of these strands. There is some overlap, and there's also some cases where there isn't overlap. So it really is just random copying of your DNA that's going to end up in your new molecules of DNA. And in order to look at this process, there was a set of scientists, and they made what's known as the famous Meselson and Stahl experiment to look at DNA replication. And what they did is they actually grew up some E. coli bacteria using different forms of nitrogen. And they had a radioactive isotope of nitrogen that had nitrogen 15. And it's basically a little bit heavier element there because it has that extra little piece. And what they found is you could grow up your E. coli in this heavy nitrogen isotopes, and then they would take their cells and basically explode them. And remember, with E. coli, we can do alkaline lysis to open up the bacteria cells, and that causes the release of the DNA chromosomes to give you that sort of snotty material when you mix your cells with the potassium hydroxide in lab. And if you take that and you spin it out under the right conditions, and we're talking about hundreds of thousands of Gs are being used to push these molecules down, they found that the normal DNA was actually a little bit lighter and would spin into the tubes and end up higher up in the tubes. But your heavy DNA that was grown with that radioactive isotope of nitrogen, it would actually travel further through this liquid matrix that they were using here, and so the heavy one would basically settle further there. And so it really was just a matter of the density of what they're looking at for these different forms of DNA here. And so they knew they could start looking at and having a way to label the new DNA that was being made, and to be able to distinguish between the old and the new DNA using this type of experimental approach. And so we can actually make some predictions like you would in a good scientific experiment. You should be able to make some predictions about what might happen with your results here. And if we take a look at the conservative model, you would be able to distinguish between the old and the new here relatively easily, because remember this idea that our DNA molecules are going to come from completely separate origin here. The old DNA molecules are going to stick back together with your two strands there, and the new ones are going to stick together over here. And so if you think about this, you're going to start out with your heavy nitrogen in your both of your DNA strands, and so you would expect this to go further down through your matrix when you do your centrifugation, and after the first round of replication you would see two distinct separate bands there. One of them with the normal nitrogen form that's going to be in the new DNA, and some of them that are left over from the previous generation there that are your heavy one. And so when they looked at this and did the actual experiment, they got an interesting little result. They got some mixed DNA there after one generation or one round of DNA replication. In this mixed form is part with the light normal nitrogen and part with the heavy. So this way they could actually rule out that conservative model of DNA replication because of these results right here. And if you think about the other models for their prediction, well, you would be able to distinguish between these two, because if you think about it, this one is just a mixture. It's going to be all over the place. But think about what's going on. After that first generation, your semi-conservative is going to look like that. It looks like a mixture. But if we go to the second generation, now we're going to start to see some differences between these two models here. And if we look at this, after growing their cells through 2 generations there, they were able to distinguish and find that that semi-conservative model of DNA replication was indeed the model that the cells were using here. After 2 generations there, they're getting the mixed band and they're getting this normal kind of DNA, or normal nitrogen in their DNA there. And so that would tend to indicate that you don't have that dispersive model, because there you would get your mixed DNA coming out after that second generation right there. And so this was the first really good definitive proof that when you're looking at DNA replication, you need to come up with a model that accounts for this semi-conservative DNA replication, where the new DNA molecule is half old and half new DNA. And so we can move on and take a look at this process that cells are using to replicate their chromosomes. And remember, when we're looking at this, this is what's known as the vertical transmission of DNA, because it's going from one generation to the next to the next. This one over here is horizontal because it's going across the horizon right there. And so if we look at this process that cells are using to replicate their chromosomes, we have to come up with, well, where do cells know where to start this process here? And that's where we can take a look at this thing right here. It's known as the origin of replication. And we can find it on prokaryotic chromosomes. They usually have 1 or 2 ories on that circular chromosome. And it's between 250 to more than 2,000 bases long. And what people have found is it's actually rich in A's and T's. And that's one of those things that makes you scratch your head. But then remember, A's and T's only have 2 hydrogen bonds. C's and G's have 3. So what the ori is really a place on the chromosome where you can unzip the 2 strands. It's a little bit easier. If you look at the ories on your eukaryotic chromosome, like this little guy, remember, eukaryotic cells have multiple chromosomes in their linear, and if you had a single origin of replication, it would work, but it turns out because these chromosomes are so much longer than the prokaryotic ones, they have multiple origins of replication, and that forms what's known as a replication bubble. And so this is going to expand in both directions here as the DNA is being copied. And this replication bubble with the little fork in the road is going to run into that next one, and that's going to end up with, there is that semi-conservative strand right there, and then the other semi-conservative strand. So you're effectively making the middle of that molecule right there to give you your 2 new DNA strands right there. And if we look at this process, I want you to be able to recognize the steps that are involved in DNA replication. And so if we look at this process here, it starts out on the origin of replication there, and you're going to have some enzymes that are known as topoisomerases, and what they're going to do is they're actually going to go in and physically break or nick the DNA strands, and that's going to help to relieve some supercoiling that you find in the chromosomes. Remember, when you look at E. coli chromosome, that thing can be stretched out to about 1 millimeter in length, but you're looking at cells that are 1 micrometer in length. So that DNA strand is 1,000 times longer than the cell. And if you take that and think about taking a rubber band and twisting it, and if you keep twisting and twisting, it's going to spiral and make that helix, and then it's going to supercoil on itself. And that's what chromosomes are doing in the cells. So this topoisomerase is basically going to cut that rubber band and allow it to loosen that supercoiling so that the DNA can be unwound for this process to continue here. after the topoisomerases relieve that supercoiling in the chromosomes, another enzyme comes in called the DNA helicase. And this enzyme is going to bind to your DNA strands, and it's going to start burning ATP for energy, and it's going to use that energy to start breaking these hydrogen bonds between the complementary bases in either strand right there. Effectively, DNA helicases are going to unzip the two strands. And remember, we're looking at a replication bubble right here. So helicases are going off to the left side, but also to the right side here when we look at that replication bubble forming there. The problem with this is DNA is not as stable of a molecule when it's in its single-stranded form, and it likes to basically reanneal the two strands and make the double-stranded form. So in order to make sure that you keep the two strands apart, because you've just spent some energy with your DNA helicase, some stabilizing proteins are going to come in here and coat both of the single strands, and these are known as single-stranded DNA binding proteins to basically prevent that reannealing from occurring there. If we look at this process, we have to go back and think about our structure for DNA. And remember, when you're looking at your DNA, it's got two anti-parallel strands. So effectively, they're going in opposite directions. But if you think about this process, your DNA helicase is chugging along here, and it's breaking those hydrogen bonds, and it's opening up both of these DNA strands right here. But these are going in opposite directions. And if you think about this process, what people found is that there is an enzyme that's going to be in cells that actually makes new DNA strands. And the problem is that enzyme can only add new bases onto that free 3' hydroxyl group on the end of a DNA strand. And so what we refer to this as DNA strands are synthesized from the 5' to the 3' end, where that hydroxyl group is. And if you think about this process, your helicase is chugging along, and so we've got this strand of DNA right there, and here we can actually make our DNA strand following along with that helicase there. And so this is what's known as the leading strand, and the enzyme known as DNA polymerase is what makes the DNA polymers. And so one of these strands is known as the leading strand because that DNA polymerase is going to lead into your replication fork, and it's going to follow along behind the helicase there. But think about this opposite strand. If that right strand is going in this upward direction, following along with your DNA helicase, the opposite strand there is going in this direction. So we can only make new DNA by adding bases onto the end of this where that free 3' hydroxyl group is. And this is a problem because now your DNA is being made in the wrong direction, because your enzyme is going to be adding bases there as that helicase is moving along. Your replication fork keeps opening, but you've got this polymerase that's chugging along in the wrong direction. And so we'll take a look at that in the next video.